Hi my dear students, welcome back to Tangal online classes. In the last class you already learned regarding monohybrid crossing over. Like monohybrid crossing over phenotype ratio as well as genotype ratio we already discussed. And today class I am going to start with the dihybridization. Dihybridization. Here, the definition of dihybridization is the crossing is made between two plants of two contrasting pairs of characters. Here, in this case, the Mendel is considered two pairs of characters are considered in one experiment. So that is called as what? Dihybrid crossing over. In the beginning, what we discuss? One experiment, one character. Now here, di means two characters in one experiment. Let's see, dihybridization means what? That is a crossing is made in between two plants of two pairs of contrasting characters. Contrasting character. It is called as what? Dihybridization. Simple is noticed or is observed two character inheritance in one hybridization. Clear? So just choose the character. Character is like the first character seed coat color. Second character is seed shape. And you know that already the seed coat color here, one character is dominant, one character is recessive. That one is yellow. Yellow color is dominant. Opposite character is green. Green is recessive. Just indicate the alliance. Capital Y for what? Yellow. Green is small y, small y. Okay. This is what? Dominant. And this is a recessive character. Okay. Now, seed shape. Seed shape also containing one pair of opposite characters. That is round seeds. And next one is wrinkled seeds. Wrinkled seeds. Round seeds genotype is capital R. Wrinkled seeds genotype is small r. And here, which is capital, it should be what? Dominant. And here this should be recessive. Yes, recessive character. Now, just take the parents. Parents are what? Always homozygous in condition. Homozygous. That is called as pure line plants you have to choose. So this character first, homozygous, round, round yellow character. Homozygous, round and yellow. So this is what? Homozygous dominant character we choose. Okay. Such plant is crossed with the which one? Again homozygous homozygous. Recessive characters are what? Green and wrinkle. Wrinkle and green. So this is actually homozygous dominant character, homozygous recessive character. Here what we are doing? We are considering two characters at a time. Two characters. One character, it is already containing opposite characters. It means how many total? 4 characters. Here, just represent the alleles of homozygous around capital R, capital R. Yellow, capital Y and capital Y. And here, homozygous round opposite. Recessive character is what? Smaller, smaller. Here capital Y is there now, right? Small y here. Just it is dominant and it is recessive. And already the Mendel is given definition for dominant. What is dominant? If the dominant genes, they can express its character in homozygous condition as well as heterozygous condition. But here we taken parents should be what? Compulsory homozygous in condition. Okay? Next. So this is which diploid cell, and here also diploid somatic cell. Next. In these diploid somatic cells undergoes meiosis division to produce haploid gametes, capital R, capital Y. Gametes. Simple. What have to do? Of diploid cells are become as haploid cells during the meiotic division. This is what? Gamete of one plant. Maybe this one is considered as a male. And here choose female plant which is containing white as well as green. Next, meiotic division is H plus, and to form small r, small y, small r and small y. Okay, so these are what? Gametes. These are what? Gametes. Now what I have to do? So these two gametes are fused. Nothing but fertilization. Nothing but fertilization. After fertilization, which zygote is formed? Capital R, small r, capital Y and small y. Zygote is formed. That is what? F1 generation. That is called what? F1 generation plant. F1 generation plant character is what? It is heterozygous. Heterozygous. Okay. So this is a dihybrid cross. Now we get the one hybrid plant, that one is F1 generation. Now what we have to do? We have to self-fertilize it with the F1 generation. Okay? Self-fertilization of F1 generation. F1 generation. Okay? In F1 generation, what type of plant is formed? Like capital R, small r, capital Y, small r. The plant is formed. So this is heterozygous seed or heterozygous embryo. So which is germinated and produced a round and a yellow color seeds, which is going to produce yellow color and a round seeds are formed. And here the dot is here, so we have capital R, small r, capital Y and small y. So here why this R gene is not expressed and why this small y is not expressed. That is already explained in Mendel. It is called as what? Law of dominant. In F1 generation, like only tall plants are formed. Here, two alleles are there, but only one allele is going to express its character. That is called as what? Dominant. And here also, this capital R, it is going to mask the or inhibit the smaller activity. And here, this capital Y also going to inhibit this activity. Okay, when smaller is expressed, when smaller is homozygous in condition, then only character is expressed. In the same, when small y is expressed, when small y's are homozygous, it means small y, in that such case only it will be expressed character. Okay, next, gamete formation. Gamete formation. So here two techniques are there. One is simple method like capital R, small r, capital Y, small r. This is a plan. Simple. So in this case actually Mendel uses a mathematical like probability is used. Here capital Y, this chances is that to fix with two times. One time capital Y and one time small y. Just write it capital R, capital Y. One gamete, capital Y, small, one gamete. Next, small r, capital Y, small r, small y. So this is actually a very simple formation of gamete. Okay. Next, one more method is there. That is here, like for example, here. Capital R and small r. 50% of capital R containing gametes have capital Y and 50% of capital R gamete are containing small y. In the same way, 50% of capital R containing gamete have capital Y and 50% of capital R bearing gametes or consist small y. So here now what going to form here? Capital R, capital Y, one gamete. Capital R, small y, one gamete. Small r, capital Y, one gamete. Last one is small r, small y. Like four gametes, four different types of gametes are formed from F1 generation. How many? Four different types of gametes are formed from F1 hybrid. Okay, in which dihybrid. In monohybrid racemose, what happened? Only two types of gametes are formed. Clear? Now what he did? For understanding of this inheritance, so he is for understanding of inheritance, he prepared one checker punnet. Like each one scale it is indicating one zygote, nothing but one seed, one plant. Like so, just make this checker punnet like. Made one checker punnet. Okay. So here is taken male gamete. Here is taken female gamete. Okay. Female plant gametes are taken. So just write it here capital R and capital Y. And next capital R small y. Next small r capital Y, and last one is small r and small. These gametes are formed. Okay. Next, same gametes are formed here only, like capital R, capital Y. capital R, small y, small r, capital Y, and small r, small y. Okay. Now simple, what I have to do? Just fuse it. So this is what? This is male pollen grain. This is female egg cell. Just we have to fuse now. So these pollen grains are going to fuse with this egg cell. Okay. Now what are formed? Zygote is formed. The zygote is which? Capital R and capital R, capital Y and capital Y. Like we have to write. And here capital, if capital letter is there, you have to write first capital letter, then only you have to write small letters. Okay. And dominant recessive genes you have to write beside. Okay. Capital R here, first capital R to capital R. Here what? Capital Y, here small y. Capital Y, small y. Like you can't write capital R and capital Y. Like we can't write. Okay. Capital R and small r. Capital Y and capital Y. Capital R, small r, capital Y, small y. So this is four zygotes are formed. Okay. Next, capital R, capital Y and small y. First we have to write capital Y. Next we have to write small y. Same you have to write like capital R, small y, small y. Capital R, small r, capital Y, small y, capital R, small y, and small y. Next, capital R, small r, capital Y, capital R, small r, capital Y, small y, small r, capital Y, and capital Y, small r, capital Y, and small y. And here, capital R, small r, capital Y, small y, capital R, small y, small y, small r, capital Y, small y, small r, small y, and small y. Okay. Now this is actually possibilities. How many plants are formed total? 4 into 4. 16 plants are formed. How many? 16 plants are formed. After formation of 16 plants, now what we have to do? Just we have to look at the character of each and every species. Just look at the each and every zygote. Here, capital R, capital Y, capital Y. It means which color? It means it is a round and yellow. Okay. Like how to write the entire characters we have to write. Next, this one is a round yellow. Here, a round yellow. How I am going to write the characters? Here, we have to consider only capital R character as well as capital Y character. We are not going to consider small r character. Why? Because it is recessive in nature. So write the character. A round capital Y is yellow. And here also a round yellow. This character also a round yellow. And here we can get the one new character, that is a round and small y is what? Capital Y is yellow, small y is green. Okay. Next, here also see what is a round seeds with yellow color, and here a round seeds with a small y means which one? Green. And in this case, a round yellow, and next here same a round yellow color. Here smaller is there. So this is what? Now the recessive genes are going to express because the smaller is not present beside the capital R. Now it can express. Smaller is what? Wrinkle character. Wrinkle and capital Y L. And smaller here same wrinkle and L. And in this case, same round and yellow, and here also be round. What you have to write? Small y character is what? Green. And in this case, here it is not round. Here why? Because smaller is there. What you have to write? Wrinkle and yellow color. And the genes here, this type of very special form, 16th plant, it is smaller, small by small. It means it is wrinkle and green. Now we have 16 plants, genotype as well as phenotype, both now we have. The next task is we have to write the phenotype ratio as well as genotype ratio. Phenotype ratio we have to notice here. Like this is 16 plants. Just what I have to consider first. This is one trait, like one zygote or one seed. So the plant is going to germinate. So the seed is going to germinate and produce round seeds with yellow color. Like visible characters, external characters like round and yellow, how many seeds are there, that you have to notice. Okay, just first this character, which one is round and yellow, round and yellow. How many characters, how many plants are there, you have to count. Round and yellow here. This is one round and yellow physical external characters we are going to consider. Second line, round and yellow. Third, round and yellow. 4, 5. This is one. 6, 7, 8, 9. So here, how many plants? Round and yellow plants. How many? 9 plants are there. Out of 16, 9 plants are what? Round and yellow. Next, we have to write possibility of genotype. Genotype of this character. Okay. Here, capital R, capital Y. Which character? Round and yellow. Homozygous. Capital R, capital Y, small. Again, round and yellow. Capital R, small r, capital Y. Again, yellow. Or capital R, small r, capital Y, and small. Again, yellow. So this is actually a possibility of a round and yellow color genotypes. Okay, such how many plants? 9 plants are found. So second phenotype, second character. Choose the second character. Where it is? Yes, this one. Around green. Just indicate A for our better identification. A. Round green. Round green. How many plants are there? Round and green. This is one round and green. This is round and green. B. But for identification, round and green. Okay. So how many? 3 plants. So 3 plants are showing what? Round and green. Okay. Then round and green alleles. A round means capital R, or capital R, small. That will give a round. For green character, it should be what? A recessive. Small y, and small y, small. Okay. Only these only possibilities. So this R should be a round and a green. Okay. That's third character. Third character is here. Wrinkle and yellow. Wrinkle and yellow, just you have to like star mark. Wrinkle and yellow. Wrinkle and yellow. How many plants are there? Wrinkle and yellow. Wrinkle, yellow, as well as wrinkle and yellow. Three plants are there. Three plants are wrinkle and yellow. And what is the character, like genotype, what have to write? Wrinkle should be recessive, smaller. No chance. Smaller, yellow. Yellow means we can write capital Y, we can write smaller, capital Y, small y also we can write. That is a genotype of wrinkle as well as yellow. Next, last character. Last one is what? Only one plant is remaining. That one is like this indicate fine. That one is what? Wrinkle and green. Wrinkle and green. Wrinkle and green. How many plants are found? Only one plant. That is, what is the genotype? Wrinkle should be smaller. Green, small. There is no alternate transfer. Okay. Now we already observed 16 plant phenotypes we observed. Just write the phenotype ratio of dihybrid cross. The ratio is phenotype ratio is 9:3:1. Here 9:3:1. Here how many plants counted? 9 plus 1, 10 plus 6, 16 plants. So this is the genotype of dihybrid plant. Okay. The next task is we have to write the genotype of dihybrid ratio. So here genotype ratio, how we are going to write? Just we have to focus on alleles, capital R, capital Y. This type of alleles, how many alleles are there in this 16 plant? So that only you have to count. For example, capital R, capital Y. Like how many plants are containing these alleles? Containing only one allele. Capital R, capital Y is only one. Like you have to count the entire 16 plant genotype you have to count. Okay. Let's write it here. The total are capital R, capital Y, capital R, small r, capital Y, capital R, capital Y, and small y, and capital R, small r, capital Y, small r, capital Y, small r, small y, capital Y, and capital R, capital Y, and capital R, small y. The last one is small r, small y, and small y. Here, so this is actually combinations of plants. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 are found. So here, this type of how many genes? Only one plant. Next, 2. So have to count. So here it is going to be a second, so that's why we can directly written. What we have to do generally, just we have to this plant, just comparing on system. Next capital small where it is there, how many is there? Like this for example, smaller. How many alleles are there? Like you have to count and write. Okay. So if you count here, the overall counted the genotype is 1 is to 2 is to 2 is to 4 is to 1 is to 2 1 is to 2 and 1. So this is actually genotype. What is the genotype of dihybridation? 1 is to 2 is to 2 is to 4 is to 1 is to 2 and 1 is to 2 and 1. This is actually the genotype of dihybrid. How many plants? 1, 2, 3, 4, 5, 6, 7, 8, 9. 8 genotypes. Sorry, 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 genotypes are formed. 9 genotypes. How many phenotypes are formed? Phenotypes are 4 phenotypes are formed. And how many gametes are formed? The gametes are 4 gametes are formed. Mostly the gamete number is equal to phenotype character number. Okay, so this is actually entire regarding dihybrid class number, phenotype ratio as well as genotype ratio. Here we can notice one character. So at the beginning we cross with between like a round yellow and a wrinkle and green plant. Actually only two characters were crossed. But at the result phenotypes are 4 phenotypes are formed. 4 phenotypes, 9 is to 3 is to 3 is to 1, like 4 different plants are formed. So further he is explaining one law of independent assortment. Okay. Law of independent assortment. Law of independent assortment is explained for only dihybrid cross, dihybrid plants. So at the beginning actually we choose 2 parents. Only we choose 2 parents. That consists like which one is a round and yellow, one parent. Next a wrinkle and a green, that is one more parent. But actually after dihybrid class we got like 9 is to 3 is to 3 is to 1. New plants are formed. What are the new plants? Here, smaller. Here one new plant is formed. And second one is wrinkle and yellow. Wrinkle and yellow and wrinkle and green. These new characters are formed. So why these new characters are formed? Further, please explain law of independent assortment. The meaning is here. If one tree or one allele is containing 2 pairs of characters, so such characters are segregated. Law of independent is telling that if one hybrid is containing 2 pairs, 2 characters, genes are there in one hybrid. For example, the hybrid is what? Like capital R, small r, capital Y and small r. So this is one hybrid. It is containing 2 pairs of characters. 2 pairs of characters. 2 pairs of characters are temporarily present in with one allele. but they are not influence each other. Here, this capital Y is not going to develop the capital Y character, and this capital Y it is not going to influence or enhancement of smaller character. These two are they present in within the one room, but they are not mixed. During the segregation, nothing but during the gamete formation, they are independently assorted and entered into the different gamete. So that is actually telling Mendel's second law, law of inheritance. Okay, just write here. The law is telling that if two pairs of traits are present, two pairs of trait, nothing but characters are present in one hybrid. If two pairs of trait, nothing but characters are present in one hybrid, they should be segregated. They should be segregated. They should be segregated independent of other pair of character. And this pair of character, this pair of genes are never mixed up. Never mixed up. It means they are not going to enhance other character. Okay? When during the gamete formation, they separately enter into different gametes. Okay? So this is actually telling that. And second, why two characters are formed? For that he is telling that round character genes as well as yellow character genes are independently inherited to the next generation. So that's why in the next generation we notice some 4 characters. Okay. So this is actually entire information regarding dihybrid crossing mode. Then if you have any doubt regarding dihybrid crossing mode, just put the message in WhatsApp. This is entire information regarding dihybrid crossing over, phenotype ratio as well as genotype ratio and law of inheritance. Thank you all of you.